In this episode, we'll cover plant cloning methods, animal cloning techniques, Dolly the sheep, and the challenges and concerns associated with cloning. Let's start with the world of plant cloning. Plants can be cloned through tissue culture, a laboratory technique using small samples of plant cells. These cells are placed in growth medium containing specific nutrients and hormones that stimulate development into complete plants. Tissue culture offers several advantages over traditional propagation methods. Plants can be produced rapidly in minimal space, grown throughout the year regardless of season, and multiplied from tiny amounts of parent material. One of the benefits of cloning is that scientists can use tissue culture to preserve endangered plant species that struggle to reproduce naturally. Commercial plant breeders employ tissue culture to mass produce desirable varieties quickly. A single exceptional plant can generate thousands of identical offspring within months, ensuring consistent quality for agriculture and horticulture. Taking cuttings represents a much older cloning method that gardeners have practiced for centuries. This involves removing a portion of a healthy parent plant, typically a stem or leaf, and encouraging it to develop roots. Cuttings provide a quick, inexpensive way to produce genetically identical plants. Many common garden plants, from roses to tomatoes, can be successfully propagated this way without specialized equipment. Both methods produce clones, organisms genetically identical to their parent. This ensures desirable traits like disease resistance, fruit quality, or flower color are preserved exactly in offspring. Animal cloning requires far more complex procedures than plant cloning. Embryo transplants allow farmers to produce multiple offspring from their highest quality livestock. This technique begins with collecting sperm from a prize bull and eggs from an elite cow. The eggs are fertilized artificially in laboratory conditions, creating embryos with superior genetics. Each embryo is then split multiple times whilst cells remain unspecialized, creating numerous identical embryos. These cloned embryos are implanted into surrogate mothers who carry them to term. The resulting calves are genetically identical to each other, though not to the surrogate mothers who birthed them. This technique can produce hundreds of elite offspring annually from a single genetic pairing. Adult cell cloning represents a more revolutionary approach, creating copies from mature organisms. This process begins by removing the nucleus from an unfertilized egg cell, essentially creating an empty cellular vessel. Scientists then extract the nucleus from an adult body cell, such as a skin or udder cell. This nucleus, containing the complete genetic information, is inserted into the enucleated egg cell. An electric shock stimulates the reconstructed egg, triggering cell division as if fertilization had occurred. Once the embryo develops into a ball of cells, it's implanted into a surrogate mother's womb. This technique famously produced Dolly the sheep in 1996. Dolly demonstrated that specialized adult cells could be manipulated to create entire organisms. Her birth revolutionized our understanding of cellular development and opened new possibilities for biotechnology. Despite technological advances, cloning presents significant challenges and concerns. The primary issue involves reduced genetic diversity in cloned populations. Genetic uniformity makes populations vulnerable to diseases and environmental changes. If all individuals share identical genes, a single pathogen could potentially eliminate entire populations lacking resistance. Cloned animals often experience serious health problems. Dolly developed arthritis at an unusually young age, raising questions about premature aging in clones. Many cloned animals suffer organ dysfunction, immune system abnormalities, and shortened lifespans. The success rate for animal cloning remains extremely low, with numerous failures for each success. These failures raise ethical concerns about animal welfare and the justification for continuing such research. However, cloning research offers potential benefits, including preserving endangered species and advancing medical understanding. Studying cloned animals provides insights into genetic disorders, aging processes, and developmental biology. Next time, we'll explore speciation, examining how species change over time.